Good evening, or good morning, or good afternoon, depending wherever you are looking and watching at us. I'm Andreas Reimann, and I'm happy to moderate this webinar of the Alliance for Pulmonary Hypertension. And I'm very happy to be joined today by a prestigious faculty, which I'm introducing in a minute. Our subject today is unraveling the genetics of pulmonary hypertension. And we will be learning both from a clinician's and geneticist's point of view, but also from a patient point of view. And this is a webinar. You can of course listen. You can ask questions, however, as well, using your channel, either the chat option in your channel, either LinkedIn or YouTube or Instagram. Just write the question, and we will come to your question at the end of all presentations. We have 3 presentations of approximately 15 minutes, 10 to 15 minutes, and after that, we still have about 15 minutes to discuss. So without further ado, I'm very happy to present today's faculty. Here is already Professor David Montagny. He's from the Department of Respiratory and Intensive Care Medicine, Pulmonary Hypertension National Reference Center of the Hôpital Bicêtre, Le Kremlin Bicêtre in France, and he is Louise Bouwman. She is chair of the PH Association in the Netherlands. And we are still waiting for Dr. Martina Prapper, who is a consultant in clinical genetics at St. George's Hospital and Royal Brompton Hospital in London. And David, I think the floor is yours for your presentation, and you are happy to share your slides. Thanks.
Thank you very much for this kind introduction, and I'm very happy to be here with you and to discuss genetics today. So I will try to share my screen. I think it will be okay. I think it's okay. You will see it. Yes, perfectly. I think so. We'll discuss about genetics of pulmonary hypertension. Clearly, I think it's something very important for the patient, but also for the family of patients, because it's probably the only way to, I will show you at the end that we can probably screen early PH and follow patient at risk. And there is a lot of consequences of genetics in pulmonary hypertension. This is my conflict of interest. I work with most of pharmaceutical labs who work in the field of pulmonary vascular disease. And I don't want to give you a lot of very difficult name of genes, but 20 years ago, we do not know any genes that predispose to pulmonary arterial hypertension. In the last 20 years, we identified at least 70 genes that are associated with pulmonary arterial hypertension. And there is some different kind of abnormalities that could lead to pulmonary hypertension. We have some genes from what is called BMP TGF-beta family, very difficult name, but that means that it's genes that control the proliferation of the vessels. And when there are some abnormalities on these genes, you are susceptible to develop pulmonary arterial hypertension. There is also some genes that are involved in channels. Channels control vasoconstriction, vasodilatation, but also proliferation of the pulmonary vascular bed of pulmonary arteries. And when you have a mutation in these genes, that could also lead to pulmonary arterial hypertension. And we have some genes that is more complicated that leads to developmental disorders. That means that the pulmonary vascular bed has an abnormal development in utero that probably predisposed to pulmonary hypertension. And that there are rare genes that are currently on it. We try to understand what is the mechanism that leads to pulmonary vascular death. So from no genes 20 years ago, we have now more than 70 genes well identified, and more than this that are currently study to understand pulmonary arterial hypertension. The most important gene and the most frequent one, the first gene that has been discovered in pulmonary arterial hypertension is BMPR2. And I will try to keep the presentation of the gene to give you an example, but probably what we said about BMPR2 is also the same for the other genes involved in pulmonary arterial hypertension. So the most frequent gene is BMPR2, and we will discuss about this gene in the next slides. What is very important when we discuss genetics is a transmission, how we can transmit the risk to the member of the family. And for quite all the genes involved in pulmonary arterial hypertension, it's what we call an autosomal dominant transmission. What does it mean? All of us have 2 copies of all genes, all chromosomes, all genes are in 2 copies except for chromosome X and Y, that is for female and male, but all the other genes are in copy by 2. And autosomal dominant transmission, what does it mean? It means that when you have one of these 2 copies abnormal with a mutation, you are at risk of the disease. That means that this patient had one normal gene, blue gene, and one abnormal, orange, so he is at risk to develop pulmonary arterial hypertension. His wife has 2 normal genes and has no risk. And how does it work for the transmission? Each parent, mother and father, give 1 copy of their genes to their children. So in this situation, there is 4 possibilities. If the father gives the normal gene to his children, these children were unaffected and has no risk of pulmonary arterial hypertension. Of course, the mother always gives a normal gene, but for 50%, in 50% of the case, the father can give abnormal genes that leads to the same situation that he has. And so these children with 1 abnormal gene and this one had also an abnormal gene and are at risk of pulmonary arterial hypertension. That is the autosomal dominant transmission. Only 1 gene on the 2 copies led to risk of pulmonary arterial hypertension. What does it mean when you have this is a family tree, and we said that male are square, female are circle, and when you have the disease, you are in black. So this patient had pulmonary arterial hypertension, 1 abnormal copy of BMPR2, for example. That means that his father and his mother has probably transmitted this abnormal gene, so one of his father or the mother are probably at risk of pulmonary arterial hypertension. That means also that his brother and sisters could have the same gene from his father or mother, so they are also at risk of pulmonary arterial hypertension. And as we discussed before, it can transmit one of these mutations to his children, and so they are at risk of pulmonary arterial hypertension. So when we identify a mutation, for example, in BMPR2 in a patient, we have also in the family some people who are at risk to develop pulmonary arterial hypertension. So we will discuss after genetic testing, genetic counseling. But you understand that it's very important to explain to all these people why it may be of interest to perform testing or to consult if there are dyspneic, to have a very early diagnosis and not wait the development of severe pulmonary arterial hypertension before to see an expert of the field. So what is BMPR2? Very easily, BMPR2, basically BMPR2 is play a role of a brake on the proliferation of the cells in the pulmonary arteries. When BMPR2 work, you have a brake and the proliferation is stopped and you have a normal pulmonary arteries. When you have a mutation in BMPR2, you have no brake and the proliferation increase and leads to pulmonary arterial hypertension, proliferation of small arteries, of cell into small arteries that finally block the blood in the pulmonary arteries. So when you lost one BMPR2, you are at risk to develop pulmonary arterial hypertension because the brake doesn't work very well. The second point that is very important and quite shared for all genetic form of pulmonary arterial hypertension is that we discussed that it's another autosomal dominant transmission. You need to have one abnormal copy to be at risk. There is a female predominance because we can probably hormonal factors may promote pulmonary arterial hypertension. So there is a female predominance. When you have an abnormal gene of BMPR2, you are more at risk if you are a female than a male. And the last point is that it's very important to understand in incomplete penetrance. What does it mean? It means that you can have this abnormal gene of BMPR2, mutation on BMPR2 of one of your two BMPR2, and you may never develop the disease. If you to have an abnormal gene or a mutation doesn't mean that you will develop pulmonary arterial hypertension. Very important point. Most of the carriers of mutation of predisposing PH genes will never develop pulmonary arterial hypertension. And this is an example of a family. You can see in black people who have pulmonary arterial hypertension, but in this family there is a lot of people who carried the mutation but who had not developed pulmonary arterial hypertension. And this is very frequent for this thing. It's probably, as I said, more frequent to develop pulmonary hypertension when you are a female, and probably pregnancy may be a risk. Hormonal factors may be also a risk as compared to male. So these things are very important. Autosomal dominant transmission, you need to have only one copy to be at risk, and incomplete penetrance. If you have an abnormal copy, that doesn't mean that you will develop pulmonary arterial hypertension. If we test all PH patients, idiopathic or familial PH patients, we found around 20% of PH patients who carried a BMPR2 mutation. Very important point is that if you test only the patient who had several cases in their family, you lost half of the patient who carried BMPR2 mutation because of the incomplete penetrance. Sometimes there is only one case in the family. That doesn't mean that it's not genetic. It means that there is other carriers who had not developed pulmonary arterial hypertension. But in proportion, that means if you test only family who had several cases, you lost half of patients carrying BMPR2 mutation. And it's very important for the relatives of these patients. Even if there is one case, I think we should do genetic testing for the patient, but also for the relatives to identify the people who are at risk in the family. Other genes that are more recently discovered that may be important. I will not go further in this, but I will show you some example. But this is the brake. The brake doesn't work and there is proliferation. But there is also now, we have identified some genes that are involved in the development of pulmonary vascular bed. And that means that during development, there is abnormalities that predispose when you are children or adult to develop pulmonary arterial hypertension. Two of them are very important, TBX4 and SOX17. I will be very short on this, but to give you information. TBX4, it's not, the gene of development are not all only limited to the development of pulmonary vascular bed. TBX4, for example, have other consequences. It plays in the development of lower limb, foot, and it plays also development of lungs. So sometimes we have now we identify some patients who have not only isolated pulmonary arterial hypertension, but also pulmonary arterial hypertension, lung disorders, and some foot abnormalities or knee abnormalities, and we can identify this kind of gene. It's very important because we should treat all these parts, bones abnormalities, and also treat the lungs, not only PH. The other example is SOX17, and SOX17 is a gene that plays a role to differentiate an artery to a vein. And when you have a mutation, the development of the vascularization of the lungs is not totally normal, and this abnormal development may predispose to PH. What is important is that so you can see a lung of a patient, and you have some abnormalities on the vein. So now with CT, we can identify the patient will probably share this mutation. What is important with the discovery of the genes, and we discover that some of patients who carried abnormality in the developmental gene, TBX4, SOX17, for example, had also heart abnormality because the genes are involved in the development of heart. And so at the beginning, when I begin to work on genetics, we consider that we should not test and search for genetic abnormalities in patients who had PH associated with congenital heart disease. Now we know that some of patients with PH and congenital heart disease are also a genetic or heritable form of pulmonary arterial hypertension, and so that change. Now we test these patients with congenital heart disease and pulmonary arterial hypertension. So this is the example of what we do in France, for example. We consider that we should propose genetic counseling for all PH patients who are considered to be idiopathic, the first case, when there is a family history of PH. When a patient had PH associated with congenital heart disease, and for another rare form of PH, that is pulmonary vein-occlusive disease. In my center, for example, all these patients were seen in genetic counseling, and we propose genetic testing. We offer genetic screening. We are lucky in France. There is no cost for the patient and relatives. And what we do what is called next generation sequencing, that we are able to test all the genes of PH in one time. So it takes around 3 to 4 months to have a result of all the genes I showed you and said there is a mutation or not. And what we do after, if we identify for this patient, for example, genetic PH, we propose genetic counseling. If the patient accepts, we propose testing to this patient. And we propose to test first-degree relatives who are at risk, the father and mother, the brother and sisters, and the children if they are adults. And we propose genetic testing. And with this step, if they accept to do the testing, we identify some people who are at risk of pulmonary arterial hypertension. For example, in this family, the father, his brother, and one of his son at risk of pulmonary arterial hypertension because they carry the same abnormality, for example, in BMPR2. What is very important for the family also is that, for example, his brother have no mutation, are not at risk, and so it's not necessary to test his children because there is no risk for his children to have a mutation. The mutation does not keep does not can be transmitted to its children. So if you know that this brother has no mutation, you said that all his children are safe and at no risk to develop pulmonary arterial hypertension. And we do after step by step. For his brother who had the mutation, we know he's at risk, and we can test his children to identify who are at risk and propose counseling and testing for these first degree relatives. So we do step by step, first degree by first degree relatives. What we can propose when we identify an asymptomatic, someone who carries a mutation, we have no disease, but are at risk. I show you that it's an incomplete penetrance. That means that some people will never develop PH, but some of them are at risk of developing PH. And so we can propose for adults, for example, in my center, to try to see, to follow the subject at risk, to screen for PH and to diagnose early if there is a development of PH. This is a program we have developed in France, but now it was considered to be, it can be proposed to all subjects who are at risk of PH. To have we don't know exactly with what is a good delay to see the patient or the subject at risk, but in this program we propose to see the subject at risk, adults who carry a mutation but have no sign, no asymptomatic, to do a clinical visit, to have a blood sample, to perform exercise testing, and to have echocardiography. If there is some abnormality, we can go to whited cat to see if there is or not pulmonary arterial hypertension. And the objective of this kind of screening program is clearly to have an early diagnosis. And we have been able to demonstrate that this kind of strategy may help to diagnose very early PH, and probably if we are able to detect PH early in the course, it's easy to treat. So to be at risk may be also a possibility to be in a screening program. The last point, I have no answer, no definite answer to what to do for children who are possibility of to be at risk of PH. It depends of national ethical laws. For example, in France, we cannot propose genetic testing for children if there is no preventive treatment. And it was exactly the case of PH. We are not able to have a preventive treatment. So we cannot propose genetic testing, but we follow the children and said if there are symptoms, they should come very quickly to a PH center to perform echo to be sure that there is no PH. And finally, the last slide is that also when we identified some people who are carrying a mutation, this is a family, a French family. You can see this family, there is several cases of PH in this family, and the black point is people who carried the BMPR2 mutation. And you see this young male carried the mutation and decided not to have children because he considered there is a risk of transmission, and he decided not to have children. And this is his cousin that had the same strategy. He knows that he carries the BMPR2 mutation, and he decided not to have children. And with new technique and pre-implantation genetic diagnosis, we propose to these two males to have pre-implantation genetic diagnosis. What does it mean? We do a baby outside the mother. We take ovula, we take spermatozoid, and we do a fetus, and we implantate only the embryo who do not carry BMPR2 mutation. And so with this program, these two males had each of them had two children, and he decided to be in this pre-implantation genetic program not for block and avoid transmission of BMPR2. So there is a lot of consequences of genetic testing, and it's very important to discuss all these points when we are in genetic counseling, because it opens a lot of questions, a lot of discussion, a lot of difficulties also. So very important point, I think. This is my last slide.
Thank you very much, and I'm happy to report that Dr. Prepa is here. Prapa, sorry, is here, and will be able to present next. Thank you very much for joining, and I'll just...
Yes, hello, and sincere apologies. I had a different time. Yes, I'm so sorry.
No problem. Andreas, are you there?
Yeah, but I unfortunately the appearance of my screen changed, so I could not unmute myself. Can you hear me now? Can you hear me?
I can hear you.
Yes, we hear you.
Thanks.
We can't see you.
I know, but because my it appears that my screen changed, so I don't know. I no longer see my my panel, so I can do anything on my settings.
Okay, no problem. Let's continue
Anyway,
with Dr.
Dr. Proper, we are very happy to have you here. You're a consultant in clinical genetics at the St. George's Hospital and Royal Brompton Hospital in London. And yeah, we are very we're looking forward to your presentation on the genetics of pulmonary hypertension.
Yes, thank you very much for the kind invitation. I think a lot of the points that I'm going to cover were to an extent covered by Professor David Montagny, but I think it would be good to reiterate some of the points, and hopefully that will make it very clear for the audience as well what we mean about genetics. So if we go on to my first slide, so the next one, essentially, as you probably know, pulmonary arterial hypertension is caused by thickening in the small arteries in the lungs with increased pressure. There are lots of different subtypes of forms of pulmonary hypertension. It could be due to other causes, for example, due to left heart disease or to chronic lung disease. And according to that classification, it's number one, the pulmonary arterial hypertension, when you have thickening of the vessels, the one that is primarily genetic, whereas the rest of the forms, we don't really have such a clear underlying genetic cause. So when we talk about genetics of pulmonary hypertension, we predominantly mean pulmonary arterial hypertension. And this can be idiopathic, which means that we haven't really uncovered the cause. It could be heritable, which means that there is possibly a family history of pulmonary hypertension or an unexpected premature death in a relative that sounds suspicious. And it sometimes could be associated with other conditions or other exposures, for example, to particular drugs and toxins, or due to coronary heart disease. And if we can move to the next slide. Essentially, what do we mean about genetics? So within our DNA, we have those little bits that look like tangled bits called chromosomes. And you see in the left-hand side of the screen, you can order them in size. So chromosome 1 is the longest, and then we go all the way down to 22. They become quite short. And we can also order them in pairs, because we have one copy of our chromosomes from our mom and one from our dad. And the first 22 pairs are the same in men and women. And the last pair is our sex chromosome. So men have an X and a Y, and females have two X's. So in this picture here, which is called the karyotype, you can see the chromosome set of a female, because we have two X's. And it is a female with Down syndrome, because if you have a look at the last row, there is a whole extra copy of chromosome 21. Now, when it comes to the genetics of pulmonary arterial hypertension, usually it's not a chromosome problem, it's a gene problem. And genes are the bits within the chromosomes. And you can see here the double helix of the DNA. And you can think of genes like a series of letters, which are DNA codes, so the letters A, T, C, and G. And what most of the genetic tests do is essentially they read those letters one by one, to see if there are any letters missing, any extra letters, or any spelling mistakes. For example, in a position of a letter G, you have an A. And when this happens, we know that sometimes this doesn't allow the genes to work as they should, and therefore they can cause a genetic condition. Now, humans, we know that we have more than 20,000 genes, and they are spread all across our chromosomes. And like chromosomes, we have most of our genes in 2 copies, one from our mom and one from our dad. And this is quite important to bear in mind, because when we find a genetic cause for pulmonary hypertension, we always have to think, was this inherited by a parent? Did it occur in a child or an adult as a new event? Because sometimes spelling mistakes in genes can happen spontaneously out of the blue. Very rarely, it could still be a chromosome problem and not a gene problem, but this is not the case. And for example, TBX4 syndrome, that I'm going to briefly mention, and I think Professor Montani also mentioned, sometimes could be due to a chromosome problem, and that's chromosome 17, where the TBX4 gene is. And you can have a small missing section that takes out that gene, and it can still cause pulmonary hypertension and other problems. And if we can please move on to the next slide. I just wanted to show you the types of genetic tests that are routinely offered for pulmonary hypertension. So it used to be a so-called Sanger sequencing, which is a single gene test that is very laborious. It used to take a lot of time to complete this test. And what this test did is, if you think of genes like books, it opened that book and read the DNA codes, all the letters, one by one, to see if you can find a spelling mistake, a missing paragraph, or a few missing words, for example. But this test was quite laborious. It took a long time to complete. Now we have a more sophisticated method, which is called next generation sequencing, which is massive parallel sequencing. And essentially, it can simultaneously look at a big number of genes. It is much faster, and it is also cost effective. So most of the individuals who have genetic testing now, they do not have a single gene tested. They either have a gene panel, which means that they can have a couple of genes of interest all read at the same time, or they can even have the whole entire 20,000 genomes looked at. And then what the people in the lab do is, we have all the results, so we have all the series of letters, but then they zoom in and they only look at the genes or books of interest. So we know that pulmonary hypertension is approximately 10 to 20 genes. So we tell the bioinformatician who's analyzing the results is, can you open those books and read them letter by letter and see exactly, have we found a spelling mistake there? Does this explain this patient's condition? And there are 2 types of broad tests. One is called whole exome sequencing, and the other one is called whole genome sequencing. The whole exome looks at important paragraphs or chapters within the book. Whole genome looks at the entire book, so from a front to back page. And if we can please move on to the next slide. This to show you the gene discovery in pulmonary arterial hypertension over the years. So it was in the year 2000 that the first gene, BMPR2, which is still the most commonly involved gene in pulmonary hypertension, was discovered. And you see, since then, with the introduction of this new genetic testing method called next generation sequencing, the number of discovered genes has increased exponentially. But if you see, not with the red line, but with a gray line, that shows the proportion of cases that were explained genetically, we still have a large chunk of them where we don't know what the underlying genetic cause is. So just because we have discovered lots of genes doesn't mean that when you do the genetic test, you will necessarily find something. And there's probably still a lot more that we need to discover in future. And if we can move on to the next one, please. This just shows you, it's 2 pie charts, as we call them, and they show you, again, the unexplained cases by genetic testing and the different types of genes. And it seems that childhood onset pulmonary hypertension is more genetic in a sense, so it's highly more likely to find something when you run the genetic test, as opposed to adult onset pulmonary hypertension, where the likelihood of picking up something is much lower unless there is a positive family history. And we can move to the next one. Thank you. Now, which genes should be examined? It's important to bear in mind that there are two types of genetic tests. It could be a diagnostic test that is done in a diagnostic laboratory, or it could be a research test. A research test may include genes that we do not 100% know if there is a definitive association between the gene of interest and the condition. And what happened is that at the moment there is a consortium, lots of work being done for a lot of different types of rare genetic conditions, where we are examining all the genes to see which ones have a definitive association with a particular condition of interest. And for pulmonary hypertension, there was a publication, very recent one, and you can see there was a big number of genes examined, and it was only a handful of them, about 10 or 11, where we definitively can say that if you have a fault in one of those genes, you are at risk of pulmonary hypertension. Some of them had a moderate score, meaning that we're not as confident, but maybe as more people are tested in the future, this may tilt over the point of saying it's definitive association. And some of them had quite limited correlation with pulmonary hypertension. So if you find a gene fault in these genes, you cannot necessarily say to a family, we have discovered why you have pulmonary hypertension, but it's something that could be of interest in future as more information comes to light. And we can move on to the next slide. Thank you. And what are the benefits of genetic testing? Of course, one of it could be the classification of the disease itself. And for example, something that was labeled as idiopathic pulmonary hypertension could be so-called PVOD, or pulmonary veno-occlusive disease. This is a disease that may not have a good reaction to certain pulmonary hypertension medication and may require early release for transplantation. So, and it is a diagnosis that sometimes can mimic idiopathic pulmonary hypertension. So unless someone has a genetic test, it may not necessarily be picked up. So that's a big benefit of the genetic test itself. Alongside the same line, accurate diagnosis and screening for other features, for example, with TBX4 syndrome, screen for skeletal abnormalities such as small patella syndrome. With other genes could be hereditary hemorrhagic telangiectasia. And of course, the number 3 very important point is family screening. And as Professor Montani probably already has shown you, there is usually a 1 and 2 risk to the next generation, because if you recall, I said that we have 2 copies of our genes. Most of these conditions are dominant, and dominant means that if you have a fault in one of the 2 copies of the gene, that is enough to cause the condition. And if you see here, you have 2 chromosomes, one in red, so that's the faulty gene, let's assume, and one in green, so the second chromosome with a normal non-faulty gene. When we have children, we split them in 2, and we randomly pass one of the 2 copies to our children. So it's a 50-50 chance. Are you going to pass on the red or the green one? And that's where that figure comes from. But it's very worth bearing in mind that we also have the so-called incomplete penetrance. And penetrance is the proportion of individuals who carry a gene fault that go on to develop the condition. And it's not 100%. So we know that 1 in 3 carriers only of a gene fault will go on to develop disease. And the likelihood is higher if you are a female as opposed to a male. We are not sure why this is the case, but it's also worth bearing in mind that just because you carry a gene fault doesn't mean that you will necessarily go on to develop the disease. If we can move on to the next one, please. This is just to show you an example of a genetic test report from the UK. So this individual had what we call a gene panel. And in the middle of the slide, you can see a list of genes, the name of the gene, and next to it you will see the percentage. So 100% that says for all of them, it means how well we could read those genes or books. And sometimes the coverage isn't great because in some of the genes you have repetitive letters written again and again, and this can mess up the test. But in this case, we can see that we had a number of genes examined and all of them were covered 100%. So it's quite a reliable test. Now, if we look at the top of the slide, you see it says the gene, so it's a fault in gene BMPR2. And DNA variant, the C dot, says 683. So in position 683, there is a missing letter there, and del means deletion. And that missing letter is enough to not allow that gene to work as it should, and therefore causing pulmonary hypertension. Next to it is set a genomic location, so it tells you in which chromosome that gene is, chromosome 2, and exactly what are the coordinates. And zygosity means whether you have the fault in both copies of the gene or only one of the two copies. And in this case, it is in one copy of the gene, and we call that heterozygous. If it was in two copies of the gene, we would call that homozygous. So this individual has two copies of the BMPR2 gene, but in one of the two, he has a fault, and we know that that fault is sufficient to not allow that gene to function as it should. And you see in red classification, it says pathogenic. That means it's a disease-causing gene change. Not all gene changes are disease-causing, and some of them can be normal variation. So it's important to be quite clear about that as well. And in the bottom, it shows you how this result is communicated in the report. So this result confirms clinical diagnosis of pulmonary hypertension, and it has implications for the rest of the family who may wish to consider genetic testing. And if we move on to the next one, I just wanted to show you an example of a family tree. So in the top, you see generation 1, which is the parents, and squares are males and circles are females. And when it's lined with a cross, it means that this individual has passed away. And in the second line, you see the affected individual in gray. So as a male who is 45 years old and has developed pulmonary hypertension with a BMPR2 associated gene fault. This individual has two children aged 13 and 8. He has a sister who is 40. And you see that little pig there, that means that his sister is pregnant. And he has a brother who is 38 and doesn't have any children. And if we move on to the next slides, first of all, what we would be inclined to do is identify whether this was inherited by a parent or not. Obviously, in this situation, we cannot test the father because he's passed away and it's very unlikely that we have stored DNA. We could test the mother. Would that help us, though? If the mother is fine at the age of 94, I don't think that it would really be very informative, even if she was found to have the BMPR2 variant or gene fault. The only difference it would make is that if the mother had it, then we would know that there would be a 1 in 2 risk for this individual's brother and sister, so for her other two children. But even without this information, we would still offer genetic testing in both of his siblings. And then the next slide, please. Should we test his children? So for the children, the same principle applies to any genetic condition, not just pulmonary hypertension. We have to respect the child's autonomy, meaning the child's ability to make decisions for their own selves. And if there is a condition that we wouldn't offer any screening in childhood, then we always say, let's wait until the children are 16 or older, and then we can see them and they can decide for themselves if they want to be tested or not. However, if it's a high-risk situation where we would offer screening to the children, then the parents essentially make the decision on their behalf, and we would test them because then we would clarify whether they do need to have an investigation of pulmonary hypertension or not. So it really depends on the specific scenario. And the next slide, please. How would you manage the sister's pregnancy? So really depends on how far along she is and how she feels about this particular gene finding. Now that we know what the gene fault is in the family, we wouldn't do a gene panel test in the sister. We would only look for the specific gene change found in her brother. And that would be a very fast test, so we could have results within a matter of weeks, and we would be able to tell her if she does carry it or not. Obviously, if she doesn't carry it herself, then there's no risk to the pregnancy. But if she does carry it, then there's a 1 in 2 chance that the baby may have the same gene change as well. Does that mean that the baby will have pulmonary hypertension? Not necessarily, because remember we talked about penetrance. Not everyone who carries gene faults goes on to develop the disease. So essentially we need to discuss with her how she feels about the pregnancy. Depending on local policy, would this be a condition that would be eligible for termination of pregnancy? Or should we just wait and test the baby after birth at some point in life? So these are the type of conversations that we would have with her. And does this result inform the brother's future family planning? Absolutely. And Professor Montani mentioned about PGD, or pre-implantation genetic diagnosis, which is where you collect sperm and eggs from both partners. You fuse them in the lab to create embryos. And after a few days, you screen those embryos' DNA to see if they have the BMPR2 gene fault or not. And you only reimplant in mom's womb the ones that don't. So that takes away the difficult decision of terminating a pregnancy, and is something that could be considered. And yeah, thank you. Now we can move on to the next slide. And finally, I wanted to show you an example of another genetic test report, which is the caveat of genetic testing, which is when we do genetic tests, the results are not always black or white. Sometimes we can get a gray result, and we call them variants of uncertain significance, or VUS in short. And here you see again a genetic test report, an individual who was found to have a gene alteration in a gene called SMAD9. And under DNA variants, you see there was a fault in position 166. Instead of a letter G, there was a letter C. And the patient is heterozygous, meaning that they have one faulty copy and one normal copy. We don't have enough evidence to say if this is disease-causing or not, and therefore we cannot offer testing in other relatives for this because it wouldn't really help us make any decisions. And your geneticist would be able to discuss this result further with you. I think we're running out of time, and this is my final slide. Oh, I just want to show you, this is my final slide. So essentially, this is the scale of how confident we are about a disease causing gene change or not. And we have a scale from blue, meaning that it is not disease causing, it's normal variation, all the way to red, meaning that it is damaging to the gene and disease causing. And the VUS, the ones that are gray, are right in between in the yellow bit. So you see there is a very wide spectrum, being confident from 10% to 90% that this might be disease causing. And this is the caveat of genetic testing. You don't always get a straightforward result. Thank you. I think this is the final slide.
Thanks very much, Tina, for this very comprehensive presentation. And close the number of presentations with Lisa Baumann. Your area actually of a BMPR2 gene, if I'm well informed, and probably the last your view on genetic pH from a patient perspective.
Yes. I hope my presentation will show up. I don't see anything. Robert, can you find my presentation? Well, doesn't matter. I can start anyways. Hello, my name is Louise. I'm 48 years old. I am married. I have two sons of 18 and 16, two dogs, and I live in the Netherlands. And I was diagnosed with PAH in 2008. Okay, well, here it is. Thank you. And well, actually, you could go next, please. Yes. I was pregnant when I was diagnosed. I had a lot of complaints, and because I got blue nails, blue lips, and blue nose and stuff, I thought it would be a good idea to go to a physician. So I went to the local hospital, and after a lot of examinations, they told me there was only one possible diagnosis left, and that was PH. And I was in a very bad condition. I was in functional class 4. I didn't know it back then, but now I know. And they told me I had to deliver my kid the other day because they wanted to do that before I went to the expert center for a right heart cath. So the other day I got contraction inducers and had to deliver my kid in a natural way with 15 liters of oxygen on my nose. And lucky enough, my kid was born healthy and nothing was wrong with him, but I was a wreck afterwards. And they brought me to a PH center the day after, and they did a right heart cath. And well, the actual diagnosis was PAH. Now, well, I think you can imagine how I was after giving birth. I was really without energy, without strength or whatever. And so any physical activities were totally impossible. And they immediately started prostacyclins per IV. So I was tied up to a pump for 24 hours a day. Can you please give the next one? And here are some pictures just to show how my journey started. In the left upper corner, that's me and my husband with my tiny little baby just after giving birth. And I don't know if you can see it, but I wear a mask with oxygen. I slept a lot with them because that was my one and only hobby almost, because I couldn't do a thing in the beginning. And slowly I made some progression. I felt better, gained a little bit of quality of life. And well, you can see that I started to feel better and could give my kids a nice life. The next, please. When I was in the expert center, they did a lot of examinations to find the cause of the PAH, but actually nothing was found. And I remember they told me about the possibility of having a gene mutation, but because I had to process so much information and devastating information, I wasn't ready to do anything with the information I got about DNA research. So I just let it slide and focused on, well, actually survival, I think, and gaining strength and try to get a better quality of life. So the first few months, I didn't think of anything else. And I think, I don't know, after 4 or 6 months, I actually thought, well, maybe I have to get back to the fact that I might have a gene mutation. And I talked about it with my physician and, of course, also with my husband and my family. And he told me about, well, a few things, brief information, actually. And he didn't refer me to for genetic counseling, actually. So I don't think they provided it in that PH expert center at that time. But I really wanted to get my blood collected because I really did want to know whether I had a gene mutation or not only for myself and for my children, but I also have a brother and he has also got children. And well, my mother has a very big family. So if it would be in her family, it would have a big consequence for the whole family. So, well, that's why I wanted it. The next slide, please. And in the meantime, before I got any results back, which they actually told me could last for 3 to 6 months. So I guess it was an old-fashioned method they used back then, 2008. So I don't know what kind of changes there were in the meantime. But I switched to another PH expert center because I was interested in participating in clinical research. And that wasn't possible in the first PH expert center where I was. So, but I forgot to ask about the results the first time I was there. And the second time I came for follow-up, I asked my physician, do you know the results of my DNA research? And he actually was a little bit shocked because he thought I already knew. And he immediately made sure I had an appointment with the clinical geneticist. And so then I already thought, oh, that will mean that I have a gene mutation because he won't send me to somebody if there's nothing wrong. So my husband and I went there and I got a lot of information about, well, actually all the information we heard before of Dr. Montani and Dr. Prapa. And also a lot of information about the chances for my children and the chances of really development of PAH, but also about health insurance and the ability of getting a mortgage. So very practical information. And also he asked a lot of questions about my family because he wanted to build a family tree like we've seen before. So he asked about sudden deaths or people with heart failure or heart diseases and that kind of stuff. And finally, I received a family letter. Oh, by the way, I have a BMPR2 gene mutation. He told me that, which is the most common one in PAH. And he provided me with a family letter for my brother and my parents. And when I left the building, I really felt very sad, actually, because I felt like I felt a sort of guilt, like I was responsible for maybe giving a lot of worries and problems in my family, and that's a really nasty feeling. But well, I couldn't do anything about it, so I just told my parents and talked to them, and also to my brother, and they decided they also wanted to get tested. Please, the next one. And so they got genetic counseling also, and afterwards the blood was drawn at them. And lucky enough, they didn't have any BMPR2 gene mutation. It went very fast because they already knew what to search for, so it didn't take 6 months, but I think around 4 to 6 weeks. I don't remember exactly, but something like that. And so that was already a big weight off my shoulders, because now not the total family was involved, but just me and, well, maybe my kids. And that was another serious thing to think about, because they were at that then 3 and 1, so very young, and what to do about that. It was also a difficult issue. So next one, please. And after many conversations and overthinking everything, my husband and I decided we did not want to get our sons tested, because, well, there's nothing to do in a preventive way. So you cannot prevent development of PAH in any way. So we thought, well, why should we decide for them? And we decided that we wanted them to be able to think about it themselves if they wanted to know. And so that was it for then, until my youngest son got some complaints at, I think, 12, 13, something like that. Because in the Netherlands, children ride their bikes to secondary school. And my son had difficulty within the 10 kilometers ride each way, and took a very long time to complete it. So he was always home late and very tired. And that worried us pretty much, because we thought, oh no, this it can be true. You can't have PAH. I don't want that. But you think of that, and well, I thought maybe I should better call my physician, and he immediately arranged an appointment with a pediatric cardiologist who did all kinds of examinations, and nothing was wrong. So that was a real big relief. And when we got home, we had a long talk with our boys, and they, I don't know, I think because of this, they thought about it, and they really decided together they wanted to know whether they had the mutation or not. So they were, I think, 13 and 15. And so they said they wanted to get tested. Next slide, please. And because we were in the lockdown because of COVID, the genetic counseling took place digitally. They were both very convinced they wanted to know. So their blood was drawn, and a local blood lab the blood was drawn in a local blood lab, because we couldn't go to the hospital. And afterwards, we just had to wait. And of course, we as parents were pretty nervous, because what if one of them has the mutation, and what if PH develops? So you're constantly worried about that, and it's whenever the date comes closer, when you know the results, it gets worse and worse, and it has a very big impact on their life. So that was a very frightening situation, but it was very strange. But we got the results also with in a digital way, and both are not carriers of the BMPR2 gene mutation that I have. So it started with me, but it also ends with me. And I can't tell you how relieved we were, because we really had a small party at home, because it's you don't want it for your children. That I am sick and have to deal with this disease is one thing, but your children, you want to have them to be able to do anything they want and to become anything they want. So it was a major relief. Next slide, please. I wanted to give you some take-home messages, because if you are diagnosed with idiopathic PAH, and it might be a long time ago, and you never thought about genetic counseling, please ask for it. And that doesn't mean you have to choose immediately if you want your DNA to be tested, but just listen to this person and let them tell about the chances and everything around it, because the rules and things are different in each country, and they can explain a lot. And talk to your friends and your family about the pros and the cons, and talk to your PH team because they can provide you with a lot of information. But primarily, take your time. Just talk to the genetic counseling people and take your time to decide. And don't ever rush. If you have any doubts, just ask more questions. Actually, that were the most important things I wanted to tell you. So I hope you enjoyed listening. And if you have any questions, please ask them now, because now you have the chance.
Thank you so much, Louise, for sharing with us. You were very open, very frank, and we had comments like this, and it was very helpful to be as detailed about your history and your experience. Thanks very much. Actually, we are running out of time, but I think if you're fine, the three members of the faculty, we could probably allow 2 questions, if that's okay to you. Dr. Montani? Okay. And Papa as well? Good. Thanks very much. Question here from Dora. I had double lung transplant due to idiopathic PH a few years after I had symptoms, and I was diagnosed with PH. Can it, because I have a mutation in case of second transplantation, it would happen?
Yeah, thank you for the question. Of course, it's very difficult to have a definite answer to this kind of question, but what we know is that it was not described, but there is recurrence of pulmonary arterial hypertension in patient carrying a mutation in pH predisposing genes. But of course, transplantation is a very difficult procedure, and sometimes there is other reasons that lead to pulmonary hypertension in this kind of patients who are transplanted. And usually it's not a recurrence of the initial disease, but it's rather a complication of the transplantation, and it can be occurred, but in different mechanism.
Okay,
we change all the lungs. So probably there is no, with the lungs who came do not carry the mutation.
If I can add to this, it's sorry. Should we go to the next question?
Yeah. No, please.
I just want to say, yeah, exactly like Professor Montani said. So it's transplantation is like a form of a gene therapy, and because if there was a genetic mutation, it wouldn't be expressed in the new transplanted lung, and therefore we wouldn't expect it to reoccur if that's the case. So the same applies with heart conditions. For example, when you have a heart transplant, you wouldn't expect someone, even if they have a mutation, to develop the condition again.
Thanks very much. So we have another question from Alena, and thanks very much for sharing her experience. And she has a question to Dr. Montani and Dr. Prapa. It's the of cases where genetic disposition of PMPR2 is detected, specifically when a heterozygous pathogenic virus is identified with the PMPR gene, which is associated with autosomal dominant primary PH. What recommendations do you have to detect early signs of the disease in their tumor? Yeah.
You want? I can begin if you want.
Yeah, if you start.
Yes, what is very difficult is that there is no clear recommendation for children. For adults, we are all agree that we should propose screening program for people who carry a mutation and are at risk of PH. For children, it's more debatable to decide if we can test all children at risk or only if there is symptoms. It depends of the ethical law in each country. For example, in France, I cannot test children asymptomatic because we have no preventive treatment. and we can also only propose echocardiography, regular echocardiography until the age of 18, and at this age, the subject can decide to do or not the genetic testing. Of course, if there is symptoms, we can do genetic testing, and it could be helpful because if there is some dyspnea, for example, it can avoid invasive procedure. If, for example, children do not carry BMP2 mutation, of course, even if we have a doubt of what is the mechanism of dyspnea, we will not perform whited card. Very important for those children. And if he carries a BMP2 mutation, it changes our mind to be sure that there is no PH. So the only advice, I think, is to be able to say that to detect dyspnea, that is usually the first signs of PH, but it could be asthma, and the most frequent cause is not PH. Unfortunately, it's asthma or something that is easy to treat. This is my kind of answer, but probably Martina may have some comments on this.
Oh, thank you. No, I'm not a pulmonologist, so I don't have much to add, but I agree if you have breathlessness, that's a red flag to think about genetic testing. I really enjoyed Louise's talk, and thank you so much for your heartfelt presentations. So you got such nice results, but I found it quite interesting for your children that, you went back and forth and then they themselves wanted to be tested. So it's always, we treat each family in a unique way, and we're very much led by the children's and the parents' wishes. So we may test children even in the absence of symptoms if parents, for example, are kept up at night thinking about it, or we just wait and watch, and if there are symptoms, then we test, or we revisit when they're 16. So there's no right or wrong answer there when it comes when to test genetically.
Thanks very much, Louise. I think it would be very appropriate if you have the last word for this session. If you could share, you made a very strong point for improving on genetic testing and for encouraging parents and patients to ask for genetic testing. What can patient organizations like yours do to foster this?
Well, at least they can give a lot of information on their websites, I suppose, about not only about genetic counseling, but also about this form of PAH, because there's not a lot information you can find easily, and it's very difficult, and it's also difficult information. So what you can find, you can't read as a normal person. Doctors can read it easily, but for the most patients, it's like going crazy when you read it because you can't understand a word of it. So it's very important that there's more information on the websites, I think.
Yeah, and thanks very much. And that's why it is so important to have the knowledge sharing platform of the potentially. And this webinar is adding to that knowledge sharing. Thanks for joining us this afternoon. And I think it was worthwhile to run over time because it was so rich and so important. I'd like to be available soon on the website. I'd like to invite you to our next webinar on September the 17th on clinical trials. Thanks very much and have a great evening.
Thank you, everyone. Bye-bye.
Thanks.